Hello and welcome to Kriya Therapeutics for a virtual tour and introduction to our manufacturing operation. We've been on quite a journey to build a capital-efficient, flexible, reliable, and fit-for-purpose operation supporting Kriya's manufacturing innovations and our exciting viral vector pipeline, which is advancing from research to clinical testing. We acquired this 51,000 square foot facility in 2020 and have since stood up analytical and development labs, a pilot plant, and a multi-product clinical GMP manufacturing operation, and all the necessary technical support spaces. Today, we will walk you through the facility and our process. I want to thank you for taking time to join us, and I hope you enjoy the tour.
When we set out to build a manufacturing facility for Kriya, our goals were clear. Our first goal was to reduce the unnecessary cost and complexity of a typical biologics manufacturing facility and distill down the requirements to accommodate only what is absolutely needed for production in a safe and compliant manner. The second goal was to integrate development and manufacturing under one roof, to eliminate communication and tech transfer bottlenecks. This clear focus resulted in a facility that meets Kriya's clinical manufacturing needs, built at a fraction of the cost and time compared to others in the industry. We achieved this in part by utilizing single-use systems in the manufacturing process. This eliminated the need for process cleaning, which reduced water usage by over 90% and completely eliminated the need for clean steam. Instead of making water, we purchase it, as needed, in ready-to-use drums. We collect the small amount of liquid waste, which is treated off-site by our waste partner. Water and waste systems take up a lot of space and are complex and costly to install, validate, and maintain. Eliminating these systems enabled us to focus on our core objectives, the vertical integration of process development, pilot operations, and vial-to-vial manufacturing under one roof. We were even able to integrate on-site analytical development and quality control labs for rapid methods development and in-house QC testing. In all, the site can support about 130 full-time staff and can manufacture up to 3 products simultaneously, which is a remarkable capability in such a small footprint.
Our Kriya manufacturing operation is fully integrated. With respect to the product development lifecycle, we have all the capabilities that are required to move a product from early concept through large-scale production. We scale up our process from the bench at the 50-liter scale within our pilot plant and at the 50, 500, and 3,000-liter scale within our GMP facility. Our manufacturing facility is also an end-to-end facility, meaning that we start with a cell vial, expand it through cell culture, purify the impurities from purification, and then in the end, take our drug substance and fill it in another vial, our drug product vial, ultimately with the destination going to the patient. Our Kriya manufacturing platform utilizes single-use technology. This is important for 2 different reasons. One, we have the capability of creating closed systems that ensures process and patient safety. 2, it provides us flexibility. Our operations can adapt to every program as small changes are made. Welcome to the GMP manufacturing space. This is the main corridor behind me. In this space, we have 4 different suites. We have Production Suite 1, which is for drug substance, a Production Suite 2, which is very similar in capability and design to Production Suite 1, a filling suite for drug product filling, and a solution prep area to help support all these operations. Our GMP manufacturing facility is designed with controlled and classified spaces. To enter each of the suites, there is a specific gowning pattern that must be followed. Likewise, the materials and products, the flows are controlled within this space. In addition, each of the clean rooms is monitored for the presence of contamination.
The entire Kriya process begins with this humble cell vial. It has about 1 million cells that we will thaw in the biosafety cabinet, add to a shake flask with culture media, where it'll grow for a handful of days. Every few days we will come in and split it to a larger container with more media. This is us exponentially increasing our culture volume so that we're ready to take this out to the bioreactor to further expand the culture. Now that we've come out of the inoc room with enough culture to enter the bioreactor, we will inoculate the reactor and allow it to grow for a few days until we're ready to make the magic happen. We'll introduce transfection reagent and plasmids so that viral vector can be produced by the cell culture. After enough time has passed and we have all the viral vector produced, we will remove it from the cell by introducing a lysis cocktail and then passing it through a series of filters to collect the harvest filtrate. The harvest filtrate material is then further processed through a chromatography step where the viral vector is captured on the chromatography resin. We will then recover that viral vector after all the impurities have been cleaned off of it. This leaves us with empty and full viral vector. We will take this through a second chromatography step that will remove the empty viral vector and leave us with just full viral vector. We're kind of platformed on Acta's and Cytiva's chromatography across all of our scales from our research labs up to our largest scale of manufacturing. We will move the process into tangential flow filtration, where we are able to exchange the buffer into final formulation buffer that is both good for shelf life and good for injection into a human, and we will achieve the final concentration for the therapy. This is the end of our drug substance process.
After we've manufactured drug substance in either production suite 1 or suite 2, the material is transferred here to our filling suite. Behind me is our fully enclosed gloveless isolator with a robotic filling system. In here, we take our drug substance, filter it, and put it into the final format, which is a vial of varying sizes depending on the program. After the vials are filled, they're visually inspected, labeled, and then transferred into our cold storage area in our warehouse.
This concludes the virtual introduction to Kriya's end-to-end manufacturing operation. We hope you enjoyed the tour. Thank you for watching.